Hello, my name is Akriti, Bhattarai, and I'm here to talk about my work on identifying and classifying disease-resistant genes in white pines. The white pines comprise the subsection Strobus of the genus Pinus, with species native to Asia, Europe, and North and Central America. There are 6 species native to the U.S., and these are all incredibly ecologically important species, especially those that are found in the high elevations of the western mountain ranges. Well, why do we care about white pines? Well, all 6 species native to the U.S. are susceptible to a disease known as white pine blister rust. It is caused by a fungal pathogen, Connecticut ribicola, and it was introduced to North America in the early 1900s, but has since spread throughout the native range of most of the white pines. In combination with changing climates, increasing fires, and pests such as the mountain pine beetle, this is devastating western white pine species. Plants are completely defenseless in the face of disease. They have genes that allow them to detect and respond to pathogens. These are known as NLR, or nucleotide-binding leucine-rich repeat receptors. These have a very characteristic structure composing of an N-terminal domain, a nucleotide-binding domain, and a leucine-rich repeat. And the N-terminal domains can be comprised of the RPW8 TIR or coil domain. In order to identify disease resistance genes, first I needed to identify the gene space. So I assembled the transcriptomes of 22 white pine species. Many of the resources came from this paper, Jin et al. This also shows the native range of these species. So here's a phylogenetic representation of the species, and the numbers in black and blue represent the relative resistance ranking based on 2 different studies, with one being the most resistant and the higher number being less resistant. And as you can see, the North American species are relatively less resistant to white pine blister rust. So I used 3 programs to identify potential NLRs from the translated proteins and their coding sequences. I then filtered and combined these results to produce a more complete and representative set of predicted NLRs. I then classified these NLRs into the subfamilies based on their N-terminal domain. I also identified complete NLRs as having all 3 domains, the N-terminal, NB-arc, and leucine-rich repeat. In total, I identified over 6,000 potential NLRs. However, roughly half of these were unable to be classified because they didn't have an N-terminal domain, and less than a fifth of all of the NLRs were considered complete. This shows some of the challenges with fragmentation while working with the transcriptomic resources. Of the NLRs that could be classified, a majority of them were of the TNL type, which is expected as it is the largest subfamily in most plant species. Interestingly, the proportion of RNL type to CNL type NLRs in the Asian species was greater, whereas the proportion of CNL type to RNL type was greater in the North American and Eurasian species. And going forward, this will be really interesting to look at, specifically at the RNL genes that might be shared across the Asian species, and looking into like orthologous groups and alignments of these. Thank you for listening, and I'd just like to take a moment to thank my mentor, Dr. Wilkerson, my colleagues in the Plant Computational Biology Lab, as well as the UConn Office of Undergraduate Research for supporting this through the SURF grant.